Welcome. Today we'll explore how transmission electron microscopy, or TEM, has revolutionized our understanding of heart diseases. Since its invention in the 1930s, TEM has become an essential tool for visualizing the ultrastructure of cells, especially in cardiac research. Over the past 2 decades, its use in cardiovascular studies has soared, allowing scientists to observe changes at the nanometer scale. This technique helps us see the intricate details of heart cells, including mitochondria, sarcomeres, and more. As we journey through this video, you'll discover how TEM enables both qualitative and quantitative analyses, offering insights into heart development, aging, and disease. Let's dive into the world of cardiac ultrastructure and see why TEM is so valuable. TEM is widely used in cardiovascular research to study heart development, disease progression, and cellular signaling. Researchers have published thousands of studies using TEM to examine how heart cells change during conditions like heart failure, myocardial infarction, and diabetic cardiomyopathy. TEM allows for the detailed visualization of subcellular structures, such as mitochondria, lysosomes, and gap junctions. By providing high-resolution images, TEM helps scientists understand the underlying mechanisms of heart diseases and evaluate the effects of genetic or pharmacological interventions. This powerful imaging tool continues to shape our knowledge of cardiac health and pathology. TEM provides a window into the heart's microscopic world. With this technology, researchers can observe sarcomeres, mitochondria, lipid droplets, glycogen deposits, and more. For example, TEM images reveal how mitochondria are distributed around lipid droplets, or how the sarcomeric structure is organized in healthy versus diseased hearts. These images also help quantify the number, size, and area of organelles like mitochondria and lysosomes. By scoring the integrity of structures such as crystal and matrix remodeling, scientists can assess cellular health and detect early signs of cardiac dysfunction. This level of detail is crucial for understanding how heart diseases develop at the cellular level. Preparing heart tissue samples for TEM is a meticulous process. Freshly isolated cardiac samples, often from the left ventricle, are quickly rinsed and fixed to preserve their structure. Proper fixation is crucial to avoid artifacts that could distort the images. The tissue is then dehydrated, embedded in resin, and sectioned into ultra-thin slices. These slices are stained with heavy metals to enhance contrast, allowing for clear visualization of subcellular details. Each step must be carefully controlled, as errors can introduce artifacts that mimic or obscure real cellular changes. Mastery of sample preparation ensures that TEM images accurately reflect the true state of heart tissue. Artifacts are unwanted alterations that can arise during sample preparation or imaging, potentially leading to misinterpretation. Common sources include improper fixation, inadequate washing, or excessive staining. For example, chemical contamination can create speckled areas, while poor resin infiltration may cause tears or holes in the tissue. High beam intensity during imaging can even break or curl the tissue. To minimize these issues, researchers follow strict protocols, use freshly prepared reagents, and work closely with trained pathologists. Recognizing and avoiding artifacts is essential for producing reliable, high-quality TEM images that truly represent cardiac ultrastructure. Once high-quality TEM images are obtained, analysis begins. This involves both qualitative and quantitative assessments. Qualitatively, scientists look for structural changes such as disrupted mitochondria or altered sarcomeres. Quantitatively, they measure the number, size, and area of organelles using specialized software. Consistency is key. Images must be taken from representative regions and at the same magnification. At least 10-20 images per sample are recommended to ensure accuracy. While TEM excels at providing detailed 2D images, it has limitations in capturing 3D structures, which sometimes requires additional techniques like electron tomography. TEM has revealed how heart diseases alter cellular ultrastructure. In conditions like hypertrophy, hypertension, and heart failure, TEM shows disorganized sarcomeres, damaged mitochondria, and increased lipid droplets. These changes are seen in both human and animal models. For example, in dilated cardiomyopathy, TEM detects smaller, aggregated mitochondria and disrupted myofilaments. The presence of autophagic vacuoles, lysosomes, and glycogen granules is also commonly observed. By comparing healthy and diseased tissues, TEM helps identify cellular markers of disease progression and provides a foundation for developing targeted therapies. Mitochondria are central to heart cell energy production, and their morphology changes significantly in disease. TEM studies show that in heart failure and cardiomyopathies, mitochondria often become smaller, fragmented, or swollen with disrupted crystal. These structural changes are linked to impaired energy metabolism and increased cell death. Quantitative TEM analyses measure mitochondrial volume, area, and density, providing objective data on disease severity. Understanding these mitochondrial alterations is key to unraveling the mechanisms of heart dysfunction and identifying new therapeutic targets. Autophagy is the process by which cells degrade and recycle their components, while mitophagy specifically targets damaged mitochondria. TEM is invaluable for visualizing autophagosomes and mitophagosomes in heart tissue. In heart diseases, increased numbers of these structures indicate heightened cellular stress and attempts at self-repair. Studies have shown that both autophagy and mitophagy are upregulated in response to pressure overload, ischemia, and metabolic disturbances. By quantifying these processes, TEM helps researchers understand how the heart copes with injury and identifies potential points for therapeutic intervention. Ischemic heart diseases, such as myocardial infarction, cause profound ultrastructural changes. TEM reveals disrupted myocardial fibers, smaller and rounder mitochondria, and increased autophagosome numbers. These changes reflect the heart's response to oxygen deprivation and subsequent tissue damage. Studies using TEM have also examined the effects of treatments like resveratrol, which can increase autophagic vacuoles and lysosomes, potentially aiding in tissue recovery. By providing detailed images of these changes, TEM helps researchers evaluate the effectiveness of interventions and understand the cellular basis of ischemic injury. Metabolic conditions like diabetes and obesity also impact heart ultrastructure. TEM studies show increased lipid droplets, fragmented mitochondria, and altered glycogen levels in diabetic hearts. These changes contribute to lipidexicity and impaired cardiac function. Animal models have demonstrated that exercise can mitigate some of these ultrastructural abnormalities, highlighting the importance of lifestyle interventions. By documenting these changes, TEM provides a clear link between metabolic health and cardiac cellular integrity. Aging is associated with distinct changes in heart ultrastructure. TEM analyses reveal increased autophagic vacuoles, dysregulated gap junctions, and the appearance of enlarged, elongated mitochondria, sometimes called megamitochondria. These changes are linked to reduced cardiac function and increased risk of disease. Interestingly, interventions like caloric restriction or spermidine supplementation have been shown to restore some of these structural features, suggesting potential strategies for promoting healthy cardiac aging. TEM is the gold standard for visualizing autophagy in heart tissue. It allows researchers to directly observe autophagosomes, lysosomes, and their interactions with mitochondria. In experimental models, genetic manipulation of autophagy-related genes such as ACT7 or Parkin leads to observable changes in autophagic structures. For example, Parkin knockout mice show fewer mitophagosomes and increased mitochondrial damage. These findings help clarify the molecular pathways involved in cardiac autophagy and guide the development of targeted therapies. Mitophagy is a specialized form of autophagy that removes damaged mitochondria, maintaining cellular health. TEM studies have shown that proteins like PINK1 and Parkin are essential for this process. In heart disease models, deficiencies in these proteins lead to accumulation of dysfunctional mitochondria and worsened cardiac outcomes. By visualizing mitophagosomes and quantifying their numbers, TEM provides direct evidence of mitophagy activity and its importance in cardiac health. The balance between mitochondrial biogenesis, fission and fusion determines the size, number, and health of mitochondria in heart cells. TEM studies have revealed how genetic alterations in proteins like PGC1 alpha, DRP1, MFN1, and OPA1 affect mitochondrial morphology. For example, overexpression of PGC1 alpha leads to enlarged mitochondria, while DRP1 knockout results in elongated, interconnected mitochondria. These structural changes have direct consequences for cardiac function and energy metabolism. TEM is also used to study lipid droplets, LDs, in heart cells. In metabolic diseases, increased LD size and number are observed, often in close association with mitochondria. This accumulation contributes to lipidexicity and impaired cardiac function. Studies in animal models, such as ATGL knockout mice, have shown that disruptions in lipid metabolism lead to structural and functional cardiac abnormalities. TEM provides the high-resolution images needed to quantify these changes and understand their impact. Recent advances in TEM technology have expanded its capabilities. Techniques like 3D TEM, cryo-EM tomography, and correlative light and electron microscopy, CLEM, allow for more detailed and accurate visualization of cardiac ultrastructure. Immunogold labeling enables the identification of specific proteins within cells, providing molecular context to structural observations. These innovations are helping researchers overcome traditional limitations of TEM and gain deeper insights into heart disease mechanisms. While TEM is a powerful tool, it has its challenges. Sample preparation is time-consuming and requires expertise to avoid artifacts. TEM provides only a snapshot in time, making it difficult to study dynamic processes like mitophagy flux. Additionally, the technique is costly and not suitable for high-throughput analysis. Despite these limitations, careful planning and the integration of complementary methods can maximize the value of TEM in cardiac research. TEM is most effective when used alongside other imaging and molecular biology techniques. For example, live cell imaging captures dynamic events, while TEM provides high-resolution structural details. Combining TEM with immunogold labeling or biochemical assays allows researchers to link structural changes with functional outcomes. This integrated approach offers a comprehensive understanding of heart disease at both the molecular and cellular levels. The future of TEM in cardiac research is bright. Ongoing developments in sample preparation, staining, and imaging technology promise even greater resolution and accuracy. The adoption of 3D imaging and advanced labeling techniques will enable more precise quantification of complex structures like mitochondria. As these innovations become more accessible, TEM will continue to play a central role in unraveling the mysteries of heart disease and guiding the development of new treatments. In summary, transmission electron microscopy has transformed our understanding of heart diseases by providing unparalleled views of cardiac ultrastructure. From revealing mitochondrial changes to tracking autophagy and mitophagy, TEM offers both qualitative and quantitative insights that drive research forward. While it has limitations, ongoing technological advances are expanding its capabilities. By integrating TEM with other methods, researchers can build a comprehensive picture of heart health and disease, paving the way for innovative therapies and improved patient outcomes.